So welcome students to this webinar. This is the 6th Palladian Symposium of Rising Scholars. Again, my name is Christina Mark. I'll be moderating today's talk. The goal of the symposium is to celebrate and showcase the hard work of our Palladian scholars, all of whom have tirelessly worked on their projects over the past couple of months. So we have, it looks like 5 presenters, if I'm correct. 1, 2, 3, 4, 5, 6 presenters. We have 6 presenters for a variety of topics today. Oh, it looks like we lost a video. Is that right? Did we lose a video? Well, perhaps they will be able to log back in. But we have 6 presenters. A fellow Palladian mentor will be scoring each student's presentation, which will help us determine prize winners after the event. And if you have any questions, you can type them in the chat for each speaker, and we will allot 2 minutes for questions after each presentation. So again, we have 6 presenters, and you can correct me if I mispronounce your name. I am terribly sorry if I do so. I believe the first one is Aryan. Is that person here? Or did that person, I'm a friend of Aryan, okay. Oh, he has trouble logging in, someone said in the chat. Okay. Well, I suppose for the sake of timekeeping, we can move on to the next person on the list, Francesca, and we'll see if we can get Aryan to log in the meantime.
Yeah, and then I just want to premise that due to a scheduling conflict, because I have a family event after this, I just have to leave like after I present and after the questions. So if I leave and it's like you're worrying about me logging in, don't worry about it.
Okay, all right, that's fine. All right, let me know if you have trouble sharing your screen. I will mute myself now.
Yeah, I'm just having a little trouble sharing my screen. It's telling me I have to quit the Zoom. To be able to share right now.
Sorry, I didn't realize I was muted. Are you logged into the email that you use on your Collegians account? Because maybe that would cause some issues. I have 2 emails, that's why. So when I logged on to my other email, it would not let me go in initially. Oh, there you go.
It'll work now.
Great.
All right, awesome. So should I just go whenever or?
Yeah, I think you can go now.
All right. So hi everyone, my name is Francesca and I worked with my mentor for 4 months to produce an original review paper titled Human Neural Stem Cell Perilesional Transplant Potential Recovery and Penetrating Traumatic Brain Injury. So what is this and why should you care? In the United States, around 1.7 million TBI cases are reported each year. Specifically, penetrating traumatic injury contributes to the majority of firearm deaths. Currently, there are no effective treatments that go beyond mitigating secondary injury. often leaving patients with a lower quality of life with diseases such as Alzheimer's and complications such as neuroendocrine dysregulation and seizures. Stem cells have the capability to give rise to any type of cell because they're unspecialized. And while I acknowledge the ethical issues that surround stem cells due to the fact that most that are acknowledged and known are associated with growing embryos, human neural stem cells provide a hopeful new treatment option as they are located not only during embryonic development, but also present in the adult brain of all mammalian species. Human neural stem cells also have proven differentiation, mitigation of axonal damage, recapitulation of host neural pathways, migration beyond the liaison location, and achievement of 90% engraftment in primary experiments using Sprague dolly rats, presenting themselves as a perfect candidate for further research. During my research, I discovered that location was also a prominent factor in transplantation success. So I compared two methods of transplantation. I compared intralesional transplantation, which transplants the stem cells directly inside the resulting cavity, versus perilesional transplantation, which is inserted directly around the resulting cavity due to the injury. And after comparing and contrasting primary experimental models of both models, I concluded that human neural stem cells inserted perilesionally reduced liaison size, increased neural differentiation, and had 43% more engraftment than the intralesional group, which left the cavity surrounded by glial scars, unfilled, and reduced migration. So, to conclude, PTBI is a nationwide issue that could be ameliorated through the use of human neural stem cells in clinical practice. Human neural stem cells present themselves as an excellent treatment option for future patients as they've proven to cause improvements on the cellular and physical levels of Sprague dolly rats. I encourage more scientists to put hope and time and energy into this treatment to save the quality of thousands of people's lives across our globe.
Okay, that was a great talk. Does anybody have any questions? You can also type your questions into the chat. I see somebody has raised their hand in the attendees. Allowed to talk, okay.
Hello, can you hear me?
Yes.
Hey, Francesca. Hey, I was interested in what got you initially interested in this topic. But actually, first, Christina, when questions are typed in, should we type them to the host and panelist or to everyone?
I think you can type it to it. I think you can type it to everyone.
Okay, let's do it more publicly then.
Yeah. And then I guess
yeah.
if you have questions later on and another student is speaking, you can privately send the questions to students so you don't disrupt the other person's talk.
Yep, that sounds good. Yeah, Francesca, interested in the source of your interest on this topic.
Yeah, so last year I'd taken AP biology and AP psychology at the same time, which was kind of interesting. And then I was sitting in AP psych class and we were learning about, all the neuronal or just like brain injuries and types. And basically the lesson just consisted of me making a list of all of these injuries and disorders. And just like on the side, when it said like treatment, it just said, oh, like we can give medicine for the seizures, but that's it. Oh, it was just, oh, it's not curable right now. It's not curable right now. So I was like, well, there's got to be some sort of treatment option that we can come up with. And then at the same time in AP biology, I was learning about stem cells. And so I just sort of connected the dots after junior year, and that's what got me interested in this topic.
Thank you.
Yeah.
Okay, it looks like Eugenia also has a question for you that was typed into the chat.
Hi, I actually had a very similar question as far as how you came up with this idea specifically.
Yeah, so I also took genetics, which gave me a really basic understanding of stem cells, but I wanted to explore it a little bit more. And this is like a very niche area, so it took a lot of reading to come up with this sort of idea and thesis for my paper.
Yeah, it sounds great. I mean, it sounds really advanced, and it sounds like you did kind of your reading and your research beforehand before coming up with kind of a new question.
Yeah, because when I started this whole project in general, I knew I wanted to focus on a type of brain injury, and I knew I wanted to focus on a potential treatment that hadn't really been quite finalized or explored yet. So I knew I wanted to do something with stem cells, and then that led me to the transplantation route. And then, what can transplantation be used for? And that led me to penetrating traumatic brain injury. So that was sort of the sequence that I wanted.
Got it. Well, good job on your project.
Thank you.
Great. Does anyone have any other questions before we move on? Okay, great. Well, that was awesome. I really enjoyed listening to your talk. It appears that Aryan is still having some trouble logging on. So I suppose we should continue with the next student in the session and see if they get a chance to come on in a bit. Perhaps I can look for their email and see if I can assist them in logging into this session. But until then, our next speaker will be Joshua Sampson. If Joshua is here, I believe I saw their video come in before, but it might have disappeared. Well, all right, then I will have to contact 2 people and see if they're having trouble logging on. But after that, we have Sarah. So let me know if you have trouble sharing your screen and slides.
Can you see it or not?
It's buffering, but I saw the cover slide. There you go. I can see it.
All right, let me know when
You can start. Did we just lose Sarah? Interesting. There you go.
Sorry, I got logged off.
Oh, it's okay. I don't know. Something weird is happening with this room.
Yeah, 1 second. I'll just get my presentation.
Sounds good.
Just a sec, there's like a small lag.
Okay, well, while Sarah is setting up, I'm sorry if I mispronounced your name, but Aryan, correct?
Yes.
Welcome. I'm so
Sorry, I
glad.
had a little technical difficulties. I was having trouble logging.
Yeah, I think there have been a few glitches in the room in general, logging people out and not being able to log them in. But if you will, Sarah is presenting the presentation now, if you would feel comfortable going after.
Yeah, that's totally fine.
Okay, great. And let me know if you have trouble. Okay, there you go. There's a little lag in screen sharing.
I'm not really sure, but it just stops presenting.
That is weird. Let me see. Would you have to what would happen if you logged out and logged back in? I suppose you already did that.
Yeah. Okay. I think 1 second.
Okay.
Um.
Otherwise, we can contact the person who is tech support in the session.
Yeah. Okay. Does someone else want to go until I figure this out?
Sure, that's fine. We can also move on. Aryan, would you be ready to present?
Yep, sure.
Okay,
Do
great.
you want me to share my screen now?
Yes, if you can, and if you can't, we'll figure out what's going on.
Okay, I'll try my best. Can everyone see my screen?
Yep.
Okay, I guess I'll get started. Hi everyone, my name is Aryan Anand. I'm a sophomore at Watchung Hills Regional High School in New Jersey, America. And the project I'm doing with my mentor, David, is creating haptic interfaces for the visually impaired to help them experience NBA games. I want to get started by asking a couple of questions. Are any of you guys sports fans? No? Yes? Well, I can tell you, I'm definitely a sports fan. I'm a very passionate, sometimes over-passionate basketball and soccer fan. And I'm pretty sure there are many passionate fans all across the world just like me. But what if they don't have the same experience as us? That is exactly what I stumbled upon one day when I was browsing through YouTube. In this picture, we can see two passionate fans, Steven and Mike. Mike is a passionate Liverpool fan, but he's unfortunately visually impaired. Now he still attends every single game with his cousin Steven. His cousin Steven narrates the game live so Mike can have the same experiences. Now this got me thinking, what if I could provide the visual experience of sports games to blind fans, but in another way? That is where haptic interfaces come in. Haptic interfaces are devices that incorporate tactile sensations to mimic human senses and display them back to the user in real time. They're composed of actuators which convert electrical energy into mechanical, sensors to detect objects, and data processing systems that take in data from the sensors and relay the information back to the actuators. You can think of this process as an ongoing cycle. Now here we can see some examples of some haptic interfaces. As you can see, the use of touch is implicated in all of these designs to help the users feel the objects so they're actually seeing them. Now this is the exactly type of technology I'm going to be using in my design. Instead of soccer games, I decided to go with NBA games. My design will include a cart-like object on wheels. The cart will hold an Arduino Uno microcontroller, along with two motors and a buzzer. The cart will be placed on a downsized basketball court just like this, making the court small enough to be placed on a table right in front of you. My sensors will be highly advanced ball tracking cameras placed in NBA games. Based on the location of the basketball, my code will send signals to Arduino, which will tell the motors to move the cart using the wheels accordingly. The blind user will then place their two fingers on top of the cart, putting both fingers on a buzzer. The cart is supposed to imitate the basketball. The purpose of this is to substitute the visual sense for the touch sense, so the users can locate the ball more accurately and precisely. Now, I recommend pairing this experience with NBA commentary, so users will know what is going on during the match, keeping track of score, fouls, and other stoppages. Overall, this project will greatly boost a blind fan's experience in watching NBA games. Thank you for your time today.
Thank you for giving the talk. That was really interesting. Does anybody have any questions? Let's see. Ask to unmute.
I have a question.
Yes.
So Aryan, first, props to St. Peter's for repping New Jersey basketball.
Yes, thank you.
And second, so what is, if you could elaborate a little bit, what is the actual motion that the user is able to detect? Is it the motion of the ball or the players, or can you elaborate a little bit on what that touch experience is like?
So the touch experience is just the users placing their hand on a cart. The cart is taking the signals from the sensors in the arena. Sensors are just tracking where the basketball is at all times. So it's trying to just help the blind fan know where the basketball is on the court at all times, because when they're listening to commentary, the commentator is not going to tell them at every single point where the basketball is. So they can't really know like who has it, where it is, and stuff like that. So just to help them experience the game better.
Got it. So they have a better sense of specifically where on the court the ball is. Right. And the other cues would come from the crowd reaction and the announcers and that kind of stuff. Right.
That's why at the end I said it'd be best if they paired with NBA commentary, so they know like what's the score, like who shot it, and just stuff like that.
Yeah, got it. Super cool.
Thank you.
Okay.
Sorry, go ahead.
I was just going to ask if anyone had any other questions.
Oh, perfect. I was just curious, kind of on that last slide, if you could go through what some of the tools are that were kind of listed, because I didn't see a label or anything.
So are you talking about the components in the car?
Yes.
So it would be Arduino Uno microcontroller, 2 motors, a wheel, a battery, and 2 buzzers. The buzzers, I didn't really elaborate that, but the buzzers are just to know, it will just buzz when the shot is being made, and it'll hold the buzz if the shot goes in. Those are just the components.
Got it. And exactly how do each of the parts necessarily help in kind of that tactile sensation that you were kind of alluding to?
Well, each of the parts, can you elaborate your question more? I didn't really get that one.
So I think you talked about kind of, it's okay if you don't know the answer to this question too. I understand it's kind of it's an elevator pitch. But you kind of talked about kind of feeling the movement and kind of understanding how the ball game is kind of going. So maybe not tactile sensation, but how do they kind of follow the game using these tools?
Yeah, right. So the tactile sensations, I just put that in there because those are used in previous work. So those are for just like I showed before in the previous work, just to help them know different objects without actually seeing them. But this one, it's just taking on that idea and just putting on in more of just like a car. It's just like following a robot on your hands and just moving out where it is across the court. So that's where the touch comes in. So instead of the blind users, they can't see the ball. They're just using their touch to follow the ball. I hope that makes it more clear.
I see. Okay. I feel like I understood it a little bit wrong, and now
No, that's fine.
So thank you. Thank you so much.
Thank you.
All right. Thank you for that presentation. Oh, it looks like Ryan has a question below in the chat.
Yeah.
I can read it out loud. Also mute Ryan, unless Ryan wants to ask the question.
Yeah, my question is, what sensors do you plan on using, and how would you coordinate NBA cameras from the actual game and replicate that on the physical board that you're developing?
Right. So for this project, I'm just creating a CAD model and a design. The sensors, we're still looking at the type of sensors that we're going to be using. I've not really finalized that yet, but to coordinate the NBA cameras, I guess then we have to contact the NBA on taking in what kind of data they get from the sensors, converting that into some type of code, and then relay that information back to my card using Arduino.
All right, great. Thank you.
Great. Okay. Let's move on to our next presenter then to keep time. Sarah, how are we on sharing your slides? Is there still a technical issue? I can
Can you
see
see
that.
it?
Yes.
Okay. Just let me know when it loads, because I think it's loading right now.
It's loading.
Okay.
Okay, it is loaded.
All right. Let me know when I can start.
You can start now.
Okay. Greetings all. Hi, I'm Sarah Pavthiwala, and today I'll be discussing my research project with you. My research project is a review article about the implications of mutant P53, particularly in its association with ROS production and migration. I chose breast cancer to study this pathway analysis because breast cancer is a highly impactful problem, with nearly 1 in 8 women in the US potentially developing breast cancer in their lifetime. It has a great impact on a person, both physically and psychologically. So P53 guards our genome and helps maintain genomic stability, thus keeping cancer at bay. Mutations in this tumor suppressor gene are highly seen in most types of cancer. Mutant P53 is associated with gain of function abilities that allow it to promote metastasis and proliferation. There are many different methods through which P53 can mutate. Here are a few that are listed. So during oxidative stress, I chose a few pathways related to P53 for my research, one including SCS and AMPK, which function to maintain cell homeostasis. Inhibition of AMPK or its depletion is shown to increase epithelial properties of a cell and decrease the mesenchymal properties of them. This has a direct role in promoting EMT and thus migration. 1 second. Yeah, okay. Another correlation I found was associated with mTOR. A certain number of mutant P53 isoforms were seen to cause hyperphosphorylation of empty ORC1 targets, which are known to have an impact on cell growth, proliferation, and migration. So this phosphorylation can be linked with migration as well. PI3K and AKT are also shown to influence ROS production through controlling mitochondria and NOx. They are seen to also produce highly reactive species such as hydrogen peroxide. Mutant P53 is shown to have an influence on this activity as well, and the phosphorylation, which can again have a direct impact on migration. Furthermore, by hampering and coming in between all these pathways, mutant P53 isoforms are altering the balance of ROS in the cell and disturbing cellular homeostasis. This by itself can be very harmful and lead to metastasis and even migration. Further research in this field will allow for effective therapeutic strategies in order to fight cancer better. Thank you.
Great talk. Does anyone have any questions? Okay. Okay, one question.
Yes, who?
Yes.
Hey, so that was awesome. I have a question on, so does that mean, like, is there anything behaviorally a patient can do to improve their condition, or is this purely from a treatment perspective, like with the implication of the research?
Okay, so I haven't really dwelled into the implications of this, but it's more so the pathways that are altered due to ROS, due to an imbalance in the ROS production. This could, however, be used in potential treatment strategies. Yeah, but I'm more so like researching about the implications of different signaling pathways that are associated with breast cancer and how migration and invasiveness of a particular tumor can be affected due to ROS production.
Got it. Okay.
Very
Thank
cool.
you.
Yeah. Oh, I guess I have a question. So I know that in anti-cancer therapies, people use ROS treatments to like induce oxidative stress because as you mentioned, the redox homeostasis is damaging and can increase and promote different things in cancer cells like migration. Well, because of those readouts that you're looking at and those different types of abnormalities in the pathways, do you think that they could have a type of diagnostic application or
Yeah,
maybe a biomarker one?
yeah, so like with ROS production, the thing is, it also induces like apoptosis. And we know that like with apoptosis, tumor cells could technically be killed because of, so technically you would lead to like, kind of stopping the growth of cancer cells by using ROS. However, there is a certain like level a certain level in the cell, when it exceeds, it starts altering other pathways, and that will actually stop apoptosis and thus lead to all of these other alterations, which can in turn lead to migration. So for example, like the EMT, epithelial to mesenchymal transition, that will lead to the tumor being more invasive.
Okay, great. Very cool. All right, so I think that we can move on to the next presenter, and anyone else who has a question can type it into the chat. If we have time at the end, we can go back to the questions. So our next presenter is Praneet. How are you on your slide sharing? Do you have any technical difficulties, and are you ready to go?
So just give me one moment. We'll find out if I can screen share. Okay, are you able to see the slides?
Yes.
Yes, and am I audible to everyone?
Yes.
Right, perfect. Let me know when I can begin.
You can start.
Okay, hello everyone. My name is Praneet and I reside in California. Today I'll be going over my project, which is Vision Q, short for the quest for vision. So in our contemporary world, while walking around on an everyday basis, the elderly and those with visual and hearing impairments face numerous challenges. The functionality of their senses essentially affects their ability to walk and physically move. According to the National Center for Biotechnology Information, guide dogs are what are most common to solve this issue. However, according to the New York Times, the cost of training guide dogs is as high as $60,000. This study alludes to a need for a more cost-efficient solution. So what is this solution, you may ask? Vision Q. Vision Q, short for the Quest for Vision, is a novel integrated device composed of 2 Arduino microcontrollers, 2 ESP32 Bluetooth microcontrollers, a pulse sensor, a UV radiation sensor, and 3 ultrasonic sensors. These will all together serve to assist the elderly and those with visual and hearing impairments in an effective and cost-efficient way. So this is what the design project looks like. The project itself is split up into 2 main parts. On the left over here, where you see my cursor, is the smart glove, and on the right, you have the smart cane, which will both work in correlation. So let's look at the smart glove. The smart glove will have an Arduino Uno connected to an ESP32 Bluetooth microcontroller, as seen over here. Obviously, on the actual glove, instead of this breadboard, I would be using a flexible perf board, which would work similar to a breadboard. On the other hand, the same Arduino would be connected to a 3-pin pulse sensor. The smart cane would contain an Arduino Uno composed or connected to 3 ultrasonic sensors, which would be placed at 3 distinct locations on the cane. The Arduino would also be connected to a UV radiation sensor, as seen on the left over here, and an ESP32 Bluetooth microcontroller. These 2 ESP32s would essentially establish and stabilize a connection between the smart glove and the smart cane. So why these specific sensors, you may ask? Well, the pulse sensor will essentially ensure good health by detecting pulse rate from time to time. The UV radiation sensor will ensure skin safety by tracking UV radiation, especially under the California sun. The ultrasonic sensors will basically ensure physical safety by detecting bumps, obstacles, cars, and other objects, and play a little vibration along with the sound upon detection. Altogether, these sensors will ensure the safety of people every day and everywhere. The final product will be a cost-efficient device, which will serve to assist the targeted demographic in many ways. Thank you so much.
That was a great talk. Thank you. Does anyone have any questions? I have a question. I might have missed this, but how can you elaborate more on the UV sensor and how it is going to be used in terms of like an output?
Absolutely. So let me go back to the slide where I actually showed a picture of the UV sensor, just for a better idea. So here's what the UV sensor looks like. It's a very small sensor. Essentially what the UV sensor does is it receives, it has a scale from which basically it detects the radiation that inputs from the sun. And I set certain parameters regarding, okay, after this certain range of scale, this is where the radiation actually starts to become very harmful, so harmful that the person should be alerted and should probably return back home. And then I would set certain parameters where they would receive a certain vibration, a different kind of vibration, which would essentially alert them to maybe wear a hat or go under the shade. And obviously, it is the scale which would be set as parameters, which would essentially determine what needs to be done when.
Okay, cool. Does anyone have any other questions? We have time for one more before the next speaker. Okay. Well, if no one has any other questions, thank you for the talk again. And
Actually, I have one for Praneet, just similar. What's the what was kind of the initial interest in this topic for you?
So initial interest, where I reside in California, I actually live near a school where students with visual and hearing impairments essentially attend school. And oftentimes I see them, playing around at playgrounds and essentially connect with their friends. I'm also a Boy Scout. I've been there for volunteering service, stuff like that. I've also seen elderly walking around the park with guide dogs. So that sort of inspired me to look into, any other efficient methods to both cost efficient and overall just get them the feeling of the nature and walking outdoors. And that essentially allowed me to work with microcontrollers, which I already have experience with, circuitry SP32 microcontrollers. They're very efficient in terms of Bluetooth establishment, and they're also very cost efficient. So it sort of allowed me to research into how I can sort of put all of these sensors together and basically formulate a device or two devices, a smart kin and a smart glove, which they can use instead of a guide dog to.
Got it. Thank you.
all right, thanks. And I suppose our last speaker is Tara. I'm sorry if I mispronounced your name. Are you ready to share your slides, or do you have issues sharing your slides?
Let me just try sharing my screen and you can let me know if you can see it.
Okay. Yes.
Oh, okay. Let me know when I can start.
You can start now.
Okay. Hi, my name is Girisaballa, and the project I completed was the structural and functional differences in the amygdala in children and adolescents with autism and how they're associated with ASD symptomatology. I chose to do this project because I've always had a long-time interest towards neuroscience and psychology, and because my brother was diagnosed with nonverbal autism as a child. My paper investigates the size, volumetric, and growth differences in the amygdala in children and adolescents with autism compared with controls using MRI studies, the functional differences in the amygdala in children and adolescents with autism compared with controls using fMRI studies, and also discusses how these abnormalities are associated with ASD symptom presentation.
After reviewing literature, I found that adolescents with autism had significantly smaller amygdalae as compared to typically developing age and sex match controls. On the other hand, children with autism were found to have significantly larger amygdalae as compared to typically developing age and sex match controls. In both cases, however, these abnormalities contributed to certain ASD symptoms, such as a lack of theory of mind, difficulties in processing emotion from facial expressions, and other cognitive, social, and emotional impairments and deficits. These results also highlighted an atypical change in amygdala volume with time from childhood to adolescence. The amygdala in individuals with autism was also found to be either hyperactive or hypoactive. Some studies reported an exaggerated or elevated amygdala response when viewing facial expressions or doing a theory of mind task, correlating with deficits in joint attention, eye gaze, and emotion recognition. On the other hand, some studies reported a decreased level of amygdala response and functional connectivity, correlating with slower judgment of facial expressions and impairments in emotion recognition. Some postmortem studies have also revealed cellular differences in amygdaloid nuclei. An increased cell packing density and smaller neuronal size was observed in individuals with autism. In conclusion, structural and functional findings suggest that the size and response levels of the amygdala in autism are either below or above the normal typical range, and that these abnormalities are associated with more cognitive, social, and emotional deficits. This research may help to understand the neuropathology of the autistic brain and the etiology and underpinnings of certain autistic symptoms. Hence, findings can help to inform targets for medication and can be implemented in treatment interventions and therapies. Thank you.
All right, very interesting talk. Thank you. Does anybody have any questions? Yes.
Hi, Tara. I guess just a broader question. I don't know to what extent you're able to talk to it, but are there different types of autism or different types of behavior related to kind of different, I guess different types of autism that are correlated with the amygdala that is bigger than average or smaller than average?
I would say, I think the first thing about autism is that it's a spectrum disorder. That's why it's called ASD, autism spectrum disorder. So, and I think the most basic way to explain the spectrum is that there are individuals who are high functioning and individuals that are low functioning. And obviously the differences between individuals with high functioning and low functioning there must be certain cognitive, social, and emotional differences, and the severity of deficits must be different. And those could be attributed to differences in amygdala size, amygdala functioning, and amygdala volume. So in my paper, also, I do discuss a little bit about the need to investigate the differences of, such studies need to investigate differences between high functioning individuals and low functioning individuals as well, because there must be certain differences, structural, functional, and cellular.
Mm-hmm. Got it. Thank you.
I hope that answers the question.
Yes. Yeah. Thank you.
Does anyone have any other questions?
I have a question. Oh, Tara, I think or Kamal has a question. So
Yes. Okay, I've allowed Kamal to talk. Otherwise, I can also read the question on the chat. What's considered the cause of autism? I have heard parents' age could be one of the factors. Not sure if it's a myth.
I don't know exactly how to answer that question because, I think my research topic is quite specific. I've dived deeper into one very tiny part in the temporal lobe and how it contributes to cognitive, social, and emotional deficits. I don't know what really causes autism. In my paper, I just discussed how this one part, the amygdala, kind of contributes to some symptom presentation. So I can't really answer that question.
Okay. Eugenia, or did you have a question?
Yes, I did. I'm not exactly sure if you would know the answer to this question, but since we're kind of on the topic, and you talked about in your conclusions, it says it could be a target for medication. Did we study anything as far as neurochemical changes or neurotransmitter changes between the amygdala and other parts of the brain, or was this kind of a different kind of project?
I haven't really done a lot of research on medication implications. I do know that some of the findings, like some of the functional findings, especially like could help for certain neurotransmitter research and neuronal research, and especially the cellular findings and the nuclear findings. But in my paper, I also do discuss there's a lot of past research that's compared individuals with autism to individuals with severe amygdala lesions. And a lot of research has been done on individuals with amygdala lesions, show that implementing strategies of directing one's own gaze onto the eyes of others can help in the processing of emotional information from the eye region. So they did a lot of research on individuals with amygdala lesions, and they found that they pay more attention to the mouth region than the eye region, and a similar mechanism can be observed in individuals with autism. So I think using implications from those studies and research as well, and this research could help for, I would say, treatment.
Got it. Thank you.
Thank you.
And great job on the project.
Thank you.
Yeah, great talk, everybody. Wonderful talks. I think I learned a lot from your elevator pitches. And yeah, the questions were also really interesting. So thank you for taking the time to come here, present them, and listen to everyone. We are just about to end the session. And I know that Eugenia is one of the judges. I'm not sure if there's anything that I need to do in terms of passing on host responsibilities, or if I just end the session.
I think we just end the session. I think we're all good. I'm not entirely sure myself.
Right. Okay. Well, if that's the case, a bunch of other sessions are going on if people want to pop into the other lobbies and see all the other projects. So yeah, again, thank you everyone for coming today and taking the time to show us what you've been working on. Yeah, and I suppose if you have any other questions, you can email any of us or message Collegians in general. All right.
Great job, everyone.
Yeah. Well, see ya.